Kia ora, welcome to my screencast, first screencast. Hopefully it works. Sorry about the taskbar and this thing that keeps popping up. I can't get rid of them, but if you know how, let me know. Um, as always, we're starting with a quiz. So there are 6 features coming up, not 7, and they relate to the information we learnt on Monday, which was evolution of the skull. So what I want you to do is pause on each of them and think of the name of that feature, but also its evolutionary purpose, why it's significant, why we're learning it, what it indicates about its owner, perhaps. So this is number 1. So we're looking at the skull from underneath with the lower mandible removed. So this is the roof of the mouth. Number 2. Number 3. Number 4. Oh, that's dumb. I left the name on it. Oh well, still more you can think about. 5, represented by the red lines there. 6. Okay, and I'm going to go back and answer them one by one. So, foramen magnum is the name of this characteristic, or feature more, and that means to make a big hole. And it is a big hole in the bottom of the skull. So this is a hole through which the spinal cord attaches to the brain, and what it can indicate to us is whether its owner walked on 2 legs or 4. Because if you think about it, the skull is always facing forward, because that's very useful if you want to know where you're going. But the spine varies depending on your mode of locomotion. So on 4 legs, the spine is parallel with the floor and it's going to connect with the brain through the back of the skull, or closer to the back at least. And then on 2 legs, or as a biped, our head kind of sits on top of our body like a lollipop, and the spinal cord is going to enter through the bottom of the skull. So we can look at ancestors' fossils and make assumptions and inferences about their lifestyle and the way that they used to move around. This is a major characteristic that allows us to infer whether something was bipedal or not. Number 2, sagittal crest. It's a large ridge of bone to which the temporal muscles attach. So they expand from here through this hole here, which we're about to talk about, spoiler, to the mandible, the lower mandible there. And when we had a more harsh fibrous diet, we required thicker temporal muscles and they needed somewhere to attach. So as our diet moved towards easier, softer foods, the body, there would be a selection against exerting energy on developing and maintaining features we no longer needed, because there would be a selection for using that energy elsewhere. And this would lead to, survival differential and all that. So this reduced over time as our diet changed, and we actually don't even have any evidence of a sagittal crest anymore. This is the zygomatic arch. So I do wish I'd taken another picture where you could see it side on, as there's 2 main trends in the zygomatic arch that are important. Number 1, the piece of bone itself. So this is your cheekbone, if you feel above your ear kind of area, is thinner and more gracile itself. So a smaller actual piece of bone, again, less pressure from chewing or mastication, and so we didn't require it to be as large and robust anymore. But also the space behind it, so this part here, has become narrower as it doesn't have to accommodate as large temporal muscles. So go back onto the slides from last lesson and you'll see a side-on view, which would be a lot more useful than this here. Brow ridge is basically a thick, robust piece of bone that lines the eye sockets, and this was necessary when we had, again, a more harsh, fibrous diet, and the pressure of trying to chew that food would have cracked the eye sockets and they needed a lot more support. We can see that ours has reduced significantly. Number 1, our diet has changed, so there's going to be selection against wasting energy on developing and maintaining a feature like that, but also selection for a larger brain, and thus brain case, would have kind of taken up some of that space as well. This here we're looking at prognathism, which is basically a measure of the craniofacial angle. So you can see this angle here is quite severe, from the forehead down to the chin area, or I don't know, teeth area. And here we see that there's been an evolutionary reduction of prognathism towards what we call orthognathism. which is the case in humans, where the face lies almost directly below the forehead. And this is a result of many interplaying factors, one of them being the size of the masticatory load and effort arms has lessened, so we don't need such big levers. You know, when you learn about levers, the larger the load arm on a lever, the less force you need to exert to achieve the same amount of work. So chewing stuff that was really harsh required big levers. Yeah, and then over here, another feature, or selection pressure rather, would be the reduction in our use of smell, which required more of a kind of exposed snout, and also the fact that a more gracile jaw is conducive to language and speech and other things that developed later on. Last one, nuchal crest. This is basically the lumpy piece of bone at the back of your neck to which your neck muscles attach. Now, thinking about the effort it takes us to hold our head up versus that of a quadruped, which is constantly lifting their head against the force of gravity, requires a lot more muscular support, so a thicker piece of bone to which those muscles can attach. But also the expansion of our cranium would have taken up a lot of that lumpy space as well. If you didn't watch this at the beginning of the unit when we watched it, definitely watch it. This is Robert Sapolsky. He's awesome. He's on YouTube. He does heaps of lectures and stuff on YouTube about human behavioral evolution and just cool stuff like that. This is a video about what makes up that 1.1% of DNA that we have that's different to chimps. And yeah, really interesting. So he is useful. Today we are looking at endocranial evolution, which is endo being inside, cranium, cranial, so inside the cranium, aka the brain. So we're going to be able to identify and describe key features of the brain that differentiate Homo from earlier hominids. So the genus Homo is really separated from all of our other ancestors by the significance of our brain and what that meant for our evolution. So we're going to look today at overall trends in the brain, but also specific parts of the brain that shifted in ways that supported our evolution. On this slide, what I want you to do is pause in a second and try and recognize two trends in the overall evolution of the brain. So one is very apparent looking at the X-ray simulations here on the right, and one's more obvious when you're looking at the sketches in the middle here. So this is a chimp down the bottom, which again is what we're using as a proxy for our ancestors who had a lot of the same characteristics. And at the top is human, or modern human. So pause, recognizing two significant changes in the brain over time, and then come back. So hopefully you recognized number one, biggest obvious change over time is the increase in the volume of the brain itself. So here, the chimpanzee, 400 cubic centimeters is the volume of their brain on average. Ours is 1400, so that's over 3 times as large as a chimpanzee and as our earliest ancestors. So yeah, that makes a significant difference to the amount of activity that can happen in the brain. And we know that intelligence is a product of relative brain size to body size. So not necessarily how big your brain is, but compared to the body. So the body wouldn't have grown as much as the brain did over that time. So the ratio changed. Trend number 2, we're looking at the middle here, and hopefully you recognize that our brain is more wrinkly. More wrinkly. This is what we call convolution of the cerebral cortex, or cortical convolution or folding. So that actually increases the surface area of the cortex, and surface area indicates greater power for processing information, as the cortex does its work on the 1 to 2 millimeter thick sheet of neurons on the surface of the brain. The larger sheet allows for large brain areas and also the creation of new additional brain areas like those for language, which we're going to cover a little bit more later. So I want you to write down in your books under the title Endocranial Evolution the 2 overall shifts or trends, remembering that this unit is all about trends over time, so how things shifted from one to another. So the 2 overall trends in the brain throughout human evolution, one being volume increase and the other being convolution of the cortex, or just wrinkliness. You really don't have to go into too much detail. Oh, yeah. So once you've written that down, which is important, I also thought it would be important to look at practicing how you can interpret graphs, as they often supplement written information in exams. So if you could pause on the slide and basically just talk yourself through this graph and what it means, what it is saying, how you would interpret this, and we'll talk about that in a second. So as you can see from the axis labels, we're looking at brain capacity in cubic centimeters over time. So the most recent being the top of the graph. And what's important is that you recognize where the skulls are situated on this graph is not the data point you're looking at. The skulls are connected to those green lines, which is where in the timeline that species existed. Hopefully you recognize, what the length of the lines represents. If you don't, pause and think about it. The length of the lines, we're looking at how long that species existed. So where it, what's the word I'm looking for? Where it came into existence, I don't know, where it evolved, and then where it went extinct. Notice that the morphological differences in all of those skulls, they're really different and they have a lot going on. And we are going to be looking at, our ancestors, our cousins later on, and why they might be so different. So basically, these different pockets of human ancestors evolved under different selection pressures. And of course, this would have changed their physiology, their anatomy, and, how they were most well adapted to their environment and the selection pressures that they were existing under. Yep, okay. So, number 2. Again, same here. I want you to pause for a sec and just think about, what are you looking at here? What is this graph representing? So do that now, and it's good practice for your exams, and come back when you have thought about it. So we can see here that we're looking at a graph representing brain mass and body mass. So the relationship between the size of the brain and the size of the body. And there's a general trend shown by this line of best fit, this green line in the middle here. And that general trend is that the bigger the body, the bigger the brain. And the conclusion you can make here is not what might be the most apparent initially, which is that you want to be top right, always want to be at the top of the graph where you would be the smartest because you have the biggest brain mass in grams. Because remembering that intelligence is dependent on the ratio between body mass and brain mass. So above the green line actually indicates that a species, so up here, that a species has a greater than average brain size relative to its body mass. So the average being represented by this line of best fit. So actually the more top left you are, the more intelligent your body, as the brain needs more volume to control a larger body, there would be, less space left for smarts if you have a big body with the same size brain. So you can see here the same thing on the underside of the green line would be a smaller than average brain to body size. So the eel and the coelacanth here have smaller than average brains for their body size, and that's because they live very simple sedentary kind of lives. The Stegosaurus is also famously known for having a brain the size of a walnut or something, so we expect to see him down here. I also hope you recognize that the scale on the x-axis is logarithmic, meaning it goes up by a factor of 10 at each interval. So we'll go from 1 to 10, and then not to 20, but to 100. And this totally skews your initial conclusions about the data. So it is really important that we are recognizing characteristics of a graph like that. So if you have a look here, you can see that an eel has roughly 10 times the body mass of a rat. But a coelacanth, which is the same horizontal distance away to the right, it's not 20 times bigger than an eel, but 100 times. So this is just an example of how looking at a graph, it's really important that you can interpret that correctly. So I'm about to run out of time in about 5 seconds, but come back for episode 2, which is more about the brain. Bye.